Well, it's a great opportunity to speak today to Professor Charles Rice from Rockefeller University on behalf of the United States Israel Binational Science Foundation. I'm Peter Hotez, and I'm joined with my associate Alan Kravitz, and it's a great honor for me to see you again, Charlie. We've been talking for many years about viruses and infectious diseases, and we're really thrilled to have you linked today as part of the U.S. Israel BSF, and of course, Dr. Rice is the Nobel laureate this year in medicine, and we'll talk about that for his extraordinary work on flaviviruses and hepatitis C. And what we'll do is alternate with a series of questions today. And I guess I'll start out, Charlie. And you've been honored with the Nobel Prize for extraordinary work and pioneering work on identifying culturing the now curable hepatitis C virus, thanks in part to your work. And with your current U.S. Israel Binational Science Foundation grant, you're part of a team focusing on hepatitis B virus, particularly concerning the virus's role as a leading risk factor for liver cancer. Would you mind telling us what you can take away from what you've learned about hepatitis C research in order to apply it this project uh, in relationship to hepatitis B?
Yeah, Peter, a pleasure to see you and hear you again. I guess we only get to chat usually once a year for sort of another program that we're both involved in. But yeah, we really sort of switched a great deal of our emphasis now to um, hepatitis B virus. I mean, I think as you know, this is we have a vaccine, we have good chemotherapy, but there are 250 million people that are chronically infected with this virus and at risk of liver disease, including cancer and death. Our work in this area really does stem from a connection with an Israeli scientist, Amir Shlomai. And Amir contacted me about coming to the lab and wanted to sign up for a program that we have at the Rockefeller University called our Clinical Scholars Program. And this allows individuals who have been trained in medicine to come to Rockefeller and usually align themselves with a basic science lab, but also participate in learning how to do translational and clinical research. And so Amir sort of came to the lab, and I think with the goal of actually working on hepatitis C, because that's what we were known for at the time. And this was also at a time when these sort of miraculous new direct-acting antivirals that could be used to treat hepatitis C were coming to fruition. And I sort of told Amir that, well, maybe he better think about, sort of, something else besides just a singular focus on hepatitis C. And I mean, he'd actually been trained at the Weizmann in Yossi Shah's lab working on hepatitis B. And so he really began that work in my lab at Rockefeller, basically established the whole experimental system. And I think one of the things you ask about, what was sort of useful from our hepatitis C work in terms of moving into hepatitis B, because as you well know, this is a very different beast in terms of the virology and many aspects of the biology. But I think one of the things that was beneficial is that we'd spent a lot of time attempting to really create more physiologically relevant systems for studying hepatitis C. And as you know, these are viruses that infect human liver cells, hepatocytes in the liver. And these cells are notoriously finicky to work with. Once you sort of take them out of the organ and put them in culture, they de-differentiate very quickly. But we had established conditions with other collaborators to begin to work with these cells, both in vitro and in humanized mouse models, where we could actually reconstitute the mouse liver with a significant population of human hepatocytes. And so I think we've been able to, sort of use that sort of technology, which was originally developed for our studies with hepatitis C, to begin to sort of probe more into the biology of hepatitis B. But it was really the, the, the tenure of Amir sort of in the lab that got this whole program off the ground, and including, when he left, sort of training some successors, and then also with the BSF allowing us to sort of continue this collaboration um, with his.
So Amir is now back at Amir Shlomai is now back at Tel Aviv University, and you're
Yeah,
continuing now to work together. And how's that collaboration been going, working across the ocean? And
yeah, I think it's going very well. I mean, one of the aspects of that we're interested in is trying to understand more about this enigmatic protein encoded by hepatitis B, the X protein. Uh, for I don't know how it exactly got named that, but and the other thing that has come out of this isn't published yet, but was a new way of studying hepatitis B, which, as you know, is a partially double-stranded DNA virus, but which goes through an RNA intermediate, a pre-genomic RNA that's produced from the hepatitis B episome in the nucleus. And one of the things that has frustrated the field in the past has really been efficient cell culture systems for studying this virus that didn't require using techniques that were actually delivering a lot of DNA, either as virus or integrated transgenes into cells. And a project that we started on that was to actually try and get around that. And what we found is that you can actually initiate the entire hepatitis B virus replication cycle, not with DNA or a virus, but actually by making a mimic of this pre-genomic RNA that encodes all of the viral functions that are necessary to start off the virus life cycle. So this is we're also working on sort of finishing that up, but it actually allows us to begin to analyze the effects of new candidate drugs, because, as you know, hepatitis B can be controlled by these sort of nucleotide prodrugs that people are using to treat the infection. But it's similar to HIV in the sense that if you stop therapy, the virus just comes bouncing right back in most people.
right.
So the emphasis now is to try and come up with a functional cure for hepatitis B that would allow people to sort of control the virus without being on any kind of treatment regimen.
Yeah, I mean, despite the availability of the vaccine, it's incredible. Hepatitis B is still widespread, and
Well, you know this. I mean, it's, you think about these amazing advances that have been made in medicine, and I think you're probably more in tune with this than I am. But I guess in my naive basic scientist hat, you would think that when there's a significant advance, it's kind of like flipping a switch. It just, gets disseminated everywhere and becomes common practice. But that's not the case. And
yeah, I mean, I've spent my life moving vaccines from discovery into clinical trials, and now we have one going into phase 3 finally. And I realized that I just finished the easy part, and the hard part's left
yeah, well, we
left still left.
certainly saw that with hepatitis C. These sort of miraculous cocktails. And we've still got 70 million chronically infected people
Right,
around the world.
absolutely,
And
absolutely. Well, now you're also applying all of your incredible virology expertise in your lab to looking at COVID-19 as well. Can you tell us a little bit about what you've been looking at with COVID-19 and coronaviruses?
yeah, that's been quite an experience. I think we've all been, sort of in the trenches on this. Anybody that's interested in viruses has been captivated by this, and as has the public. So when we were sort of in the epicenter, of the U.S. Pandemic here in New York, and the Rockefeller University shut down in mid-March, except for those programs that were actually working directly on projects that were relevant to COVID-19 and SARS-CoV-2 infection. And I guess our efforts in that area have been maybe a little bit futuristic in terms of, what we're trying to accomplish. I mean, as you know, these viruses are obligate intracellular parasites. I mean, they need the host cell machinery in order to replicate and produce new progeny. And so what we've been trying to do, and we've been doing this not only with SARS-CoV-2, but also with three of the sort of common cold causing coronaviruses, is to try and understand more about the cellular machinery that these viruses need in order to propagate. Hopefully finding pathways or particular host gene products that are pan-coronavirus requiring. And this has been aided by, the other sort of Nobel Prize winning technology, sort of CRISPR, and the gene editing technology that really allows precise interrogation of the dependence that these viruses have on basically all of our genes. And I guess in some work that is just coming out, I think it'll be in the print version of Cell on January 7th, we did a number of these CRISPR screens and identified, sort of lots of potentially interesting host factors. But one of these was a predicted transmembrane protein called TMEM41B. And there's not a lot known about this protein, but it was interesting because we had actually identified this as a protein that was required by other members of this virus family, the sort of Flaviviridae family. So this includes viruses like West Nile and Zika and yellow fever. And this host protein is required for replication of those viruses in a variety of different cell types, including even mosquito cells. These are mosquito-borne viruses. And we had tested a bunch of other virus families to see if they might also be sort of requiring this function, and they were all negative. And then when we began sort of expanding our work on coronaviruses, we tested that family as well. And interestingly enough, the same protein is absolutely required for replication of the coronaviridie. So two very different families are, dependent upon this. And so
just by the fact that they're RNA viruses, but that's about the only thing they have in common, right? So.
Well, we don't really sort of know the sort of mechanistic basis of this at a level that we would like to. But it seems like what might be going on is that these two RNA viruses have to remodel cellular membranes in order to form their sort of replication organelles and factories. And we think that might be, sort of the common thread. Now, of course, other positive strand viruses do the same, but maybe the kind of membrane remodeling events that are required are slightly different for their, sort of replication.
But you're not finding these in measles virus or other RNA viruses, no.
No. So that was kind of a fortuitous finding. I mean,
that's really interesting.
We'd also been working on, and this was actually a project that was started back in the sort of heyday of hepatitis C. As you know, sort of in the early days of trying to treat non-A, non-B hepatitis and then hepatitis C, interferon was the cornerstone of therapies, despite its side effects and lack of efficacy in many of the treated patients. But we became interested in trying to understand, sort of what was it that interferon was doing that could actually block hepatitis C replication. And so we became interested in defining what some of the interferon regulated genes might be doing and which ones actually had antiviral effects for a particular virus group. And some years ago, we had done a screen with a library of these genes, an overexpression screen, and identified a protein called Ly6E that actually was a potent inhibitor of coronavirus entry. And so when SARS-CoV-2 sort of emerged on the horizon, we were already working on that and showed that the same gene could actually inhibit entry of SARS-CoV-2. So I guess a lot of what we're doing in this area right now has to do with understanding more of the virus host biology, and also trying to create some systems that would be useful for coronavirus studies at containment levels that aren't necessarily at the biosafety level 3 containment where we need to.
Yeah, that would be a huge advance because, donning and doffing all the PPE to go into the lab is exhausting. It really slows you down. So,
Well, and there just aren't, I think, if you really want to find out if what you're studying is relevant with respect to a real virus infection model, you're kind of stuck. And, a lot of institutions and laboratories, just don't have these kinds of facilities, and
yeah,
they're not easily created on a dime. You can't just sort of say, okay, I need a BSL-3 facility next week and
absolutely.
have it happen.
Absolutely. Alan, do you want to?
Yeah. Yeah, well, when I heard you speaking also, Dr. Rice, I mean, one of the things that went through my mind, on this, and uh, what do you say to the people who think it's like, okay, when these vaccines come out, that's going to be it. We're not going to have to deal with COVID anymore. It's because people are, you know, seeing it like the be all and end all, like everything's going to be back to normal right away. And,
Well,
the
that ain't
science
gonna,
says it's
yeah,
probably not true.
yeah, that's not going to happen, and
Yeah.
I think that, I mean, the initial results on these vaccines that are, sort of one of which has had in the United States here, the emergency use authorization approved, and we expect that will happen, sort of momentarily for the other one. I mean, these really are, I mean, this is amazing, to, go from discovery of a virus to actually having a vaccine, even under this emergency use authorization that's going into people in the same year. Amazing. But I think
Oh,
that,
by the way, so along those lines, I was talking with Susan Weiss from Penn, who's been working on coronaviruses, and we often point out, we have a COVID-19 vaccine now scaled up for production on the spike protein. We've been working on coronaviruses as well, and I often like to point out that, the vaccines didn't just appear out of nowhere. It required, decades of, understanding the biology of the coronavirus and identifying the spike protein as the weak link and making prototype vaccines. And, she points out she was working on mouse hepatitis virus, which is a coronavirus, for years and struggling to convince people that why this was important. So it really is a reminder of the importance of supporting fundamental virology
oh
research.
yeah, no, I couldn't agree more. And,
Yeah.
the coronavirus field was in danger of extinction in the 90s. There were basically just, a few diehards like Susan and some labs in Europe and a few other ones in the States that kept, working on the mouse hepatitis virus. But I guess SARS changed that, but only for a while, right? And then
Right.
MERS was kind of like I guess another primer. You could call it the first boost, and then SARS-CoV-2, sort of really. And I think the fundamental contributions of these people that have studied coronaviruses, you're so right, have really sort of informed what to do. And we got the same kinds of arguments in the flavivirus space. I remember, one of the things that I began working on while I was still at Caltech before I went to Washington University was yellow fever. And, my mom would say, well, Charlie, why are you working on yellow fever virus? I mean, we have a vaccine for that, and, we don't really have any yellow fever in the States. But, it was just curiosity driven research about this virus and this family and this incredible vaccine that had been, developed by Max Tyler in the 1930s. And that work that we did, I think, was really what put us in a position to actually make, whatever contributions we did to the hepatitis C field when that virus was really sort of identified by Mike Houghton in 1989. And it ended up being a new member of our virus family.
Mm-hmm.
So,
Absolutely.
yeah.
Dr. Rice, wanted to ask also, this is like a little bit more of a, personal, question for you. Has it sunk into you yet that you're a Nobel Prize winner. When I interviewed Amir, he said he could not imagine when he was more excited when he found out that you won the Nobel.
Well, I don't know. I was initially just irritated at being woken up at 4:30 in the morning, since I'm often, up late and, not used to having our landline ring at, 4:30 in the morning. And so my impression was, I didn't expect this. I'm certainly not the type of person who's going to be, sitting by the phone on October 5th, sort of waiting for the call. And yeah, so it's been a strange period of time, I have to say. I guess it has eventually sunk in, sort of based on seeing the announcement on, sort of the sort of Nobel Prize website that morning, and then the ensuing thousands of emails that have put me so far behind in terms of my correspondence that I may never dig myself out. But it has been, I think, a strange time for, sort of something like this to have happened in the context of this pandemic. Because we're all, masked and, social distancing, and, the sort of festivities in Stockholm are, sort of all virtual. And so that's, and I have to say that besides that, it's really just the situation that we're in with this global health situation that you're in. It's actually been a little bit difficult for me to, sort of enjoy all this sort of Nobel Prize related stuff when we've got all this coronavirus research going on, and trying to roll out these vaccines and get people to take them. I mean, that's really sort of what's been on my mind for.
Right. Well, hopefully maybe they'll do something the following year to kind of.
Yeah, they're sort of making some noises about our ability to crash the 2021 party in December.
That would be my recommendation.
Yeah. Well, you didn't even get to go to Sweden. You had to accept it at the Swedish embassy in New York, right?
Well, it was actually the consul general's residence. So it wasn't,
Okay, yeah.
I sort of, I think the Swedes felt terrible about this situation and not being able to, sort of really conduct these festivities as they have for, many, many years. And so, but I have to say, the consul general, she was just fantastic. And, but we decided, as the incidence rate of COVID-19 diagnoses were sort of creeping up in New York, that it would be, sort of a mistake, and but we had, probably a half dozen, sort of folks at the ceremony. It wasn't such a beautiful home that was built in 1910 by, I think, a family that had an interest in the newspaper, sort of business in the early days of when Park Avenue was being expanded. And so it was a nice, nice ceremony. And I was happy to, actually be able to stay close to home. I didn't really, I'm having so much engagement with our work in the lab right now. I didn't really want to, take on the risk of getting infected and then being, sort of taken out of commission. And we also had testing in place so that that everybody involved could have been tested the day before. So at least we knew we were RNA negative at least 24 hours before the event.
Excellent.
So where do you see things going with, based on your experience collaborating with an Israeli scientist, any overall sense of U.S.-Israel scientific collaborations, thoughts about that?
Well, I mean, I'm very sort of bullish on this program and the collaboration. At least for me, it's I think given rise to just a pipeline of fantastic scientists that have come to the lab to for various projects, and have either stayed in the States or often have gone back to Israel. I mean, in the case of Amir, I think he would have loved to stay longer, but he ended up getting recruited back to a chair of medicine, sort of position. So, I mean, I think that given his additional clinical responsibilities, this ongoing interaction that we have with him, which has really been continuous since he was in the lab and went back to Israel, has been important for both of us, both for his program and for our program. And I also have just some spectacular, sort of new Israeli postdocs in the lab, actually another one from Yossi Shaw's sort of hepatitis B, Anna Ricardo Lax, has been in the lab for several years doing a lot of really exciting, COVID-19 related work now, and some new folks as well. So I think it's a very vibrant program, and it's certainly been wonderful for my research program. And I think it's also been, hopefully Amir feels this way, good for him as well. Although, there are a few of the hepatitis B projects that did get, we were unable to actually sort of work on all this other stuff that we were doing that was non-coronavirus related for about 6 months. So we're a little bit behind on getting some of that work finished up and published. But anyway, I think things are, I hope the program will continue, and that these positive results and relationships that are established will bode well for the next decade.
Just FYI, when I interviewed Amir, he said that every time he goes to your lab, Dr. Rice, he learns so much, not only from you but from your whole team. So
well, yeah,
the
I would
very
emphasize
mutual.
that I probably don't have much to teach people anymore. So
I don't know.
I kind of
He
feel
would beg to disagree with that.
it's funny because one of the things that sort of this prize has emphasized to me is you kind of go back and think about, at least in our case, things that were happening 20 years ago, 30 years ago. When you think about the advances that were made in non-A, non-B hepatitis and hepatitis C that actually took decades to accomplish, and think about how quickly this could be done today, it's a little bit embarrassing to be giving these Nobel lectures. And I'm sure there's a whole contingent of young scientists out there who are going, what? These guys are getting a Nobel Prize for this? We could do this in a couple days.
Yeah, I remember a good part of my doctoral thesis at Rockefeller was cloning a gene.
Yeah, exactly.
That was a big deal in 1984, 1985, and now we do it in a day, right? So
yeah, no, it's amazing how science has advanced, and I think you make a good point that a lot of the advances that or a lot of the technology that we have today and its success does rest on a great body of prior work. And but I am amazed at what these young scientists have to use to the CRISPR technology.
the technology has become so powerful. One of the things that I'm very concerned about, though, and I guess we'll finish up, is this parallel and aggressive rise in the anti-science, and not only the anti-vaccine movement, but what's now become sort of an anti-science initiative or a confederacy where people actively seek to discredit masks or social distancing or create pseudoscience around fake concepts of herd immunity. And I'm sure you've witnessed this as well. And it's not only in the US, it's moving to Europe now and even possibly Israel. Any thoughts about that?
Well, it's certainly very worrying. If you asked me about this sort of when I was, starting off as a graduate student, and we would be in this space right now, it's almost unimaginable. But I think, we really have to try and establish, at least with those that are willing to have a dialogue, we need to communicate and talk to them. I mean, I'm hoping that with what appears to be a lot of success on the horizon for COVID-19 vaccines, and that there will be an enthusiastic sort of spread of enthusiasm for that. Hopefully will spread as efficiently as the virus itself. And really sort of, if you will, sort of immunize some of the people that are, not too supportive of the vaccine. It's a little bit hard to know, sort of how to penetrate, some of these organizations that occur on social media that just sort of propagate, these concepts. So I'd be interested. I mean, you've been in the trenches on this, Peter, and in terms of link between vaccines and autism and so on, and I think we need to at least start by trying to penetrate, sort of maybe the not the sort of core non-believers, but at least those that are still wavering. And this pandemic and the response to this pandemic, and I mean, that's sort of what I was hoping with sort of the recognition of the hepatitis C sort of success story, that people would actually see that sort of science could have benefit.
Right.
For their
One of the if there's any there's not many silver linings to this pandemic, but one of the things that I've noticed is that people on the cable news networks, they like hearing from scientists, not necessarily like hearing everything filtered through the journalists or the anchors, and people are hearing from scientists directly now for the first time, and they generally like what they're hearing. They like the fact that they're not being spoken to like they're in the fourth grade or the sixth grade. They actually appreciate some of the complexity. So I think that's that's very gratifying.
Yeah.
Well, I think we're about to wrap up then, and
Okay.
incredibly grateful for your time, Charlie. I know how incredibly busy you are, and your work is so important.
I'm probably only about 10% as busy as you are.
No, not at all. Well, I think we're all busy. I think it's just the level of intensity is so extraordinary right now.
Well, but one of the other things that has taken some of my time has been this sort of clinical task force thing that sort of got established back, sort of before the election, and the Governor Cuomo here decided was a good idea, which I think has taken on its proper role, which is really more to trying to encourage people to get vaccinated for things that get approved under the EUAs. But that's actually been an interesting sort of diversion for me. I agreed to do.
Well, that's great. It's really important that you're doing that.
Back in September, but the playing field has changed very quickly, and I'm at least happy about that. And
well, thank you again for everything.
We'll look forward to seeing you on TV.
In fact, that's why I'm getting called now by MSNBC to go on it.
Well.
Okay, so we'll see you tonight then.
Well, no, you'll see me if you like, you'll see me in 15 minutes with Katie Torr. But
okay, well, keep it up. It's really important.
Thank you, and thank you for your time, and Anton, thank you for organizing this. I really appreciate it.
And thank you both, Peter and Dr. Rice, because we know you're both incredibly busy.
Thank you. I'm going to run, but thank you again, Charlie. I appreciate it.
Charlie.
Thank you.
Okay. Yeah.
Bye-bye.
Right.
Bye. Thank you, Stephanie. Thank you, Anton. Bye-bye.
Bye-bye.
Yeah. Charlie, if I could just ask, actually, it's talking about being busy, if I could just ask you, one more question, and it kind of like deals with work-life balance.
What's that?
Well, that may answer the question right away, but it's, especially Nobel laureates have this, I don't know, this kind of aura, around them. And, what can you share about work-life balance, and what advice might you have for early career scientists in that regard?
Well, I don't know. I mean, I've always found that, well, since my passion is to is science, there are relatively few things that I would, sort of prefer to be doing other than, sort of either being in the lab when I was actually doing bench work, or, sort of being in the lab hounding people who are now, sort of doing the bench work. But I mean, I do think you need some, sort of time away from your sort of work passion. I guess for me, that's varied, throughout my life. There was a time when I was a real sort of movie buff, and, very much enjoyed all kinds of different music. I guess I also have a real love of the outdoors. And so hiking and backpacking, that's something that we still try and do, for at least a break once a year to, get out of New York City and get to an extremely remote spot in the Wyoming Wind River Range. And then I also, we've also had a very dedicated canine support group. So we've had just sort of a wonderful series of dogs, sort of throughout this period of time. So I enjoy very much sort of getting outside and throwing the frisbee for the dog, and sort of playing with the dog. Then finally, this is something that was a side effect of my undergraduate training at UC Davis, but as you may know, that school is particularly well known for its viticulture and enology program, so grape growing and winemaking. And there was, after an exposure to an introductory course in that at UC Davis, I kind of became interested in that. And there was a time when I was teetering between, sort of going into graduate school and sort of on this research trajectory versus, going off and more training and becoming a winemaker. So I haven't really done that, but I do have an interest in wine, and actually have been enjoying some very nice ones from Israel. So that's kind of another, pseudo hobby of mine. Probably not the healthiest one in the world, but anyway.
Well, maybe this is a good point to wrap it all up.
Okay.
So, Dr. Rice, thank you so much for making your time available, and we are very appreciative at the BSF.
No.
And we are also very proud and privileged that we have made like a tiny contribution to your success, to these BSF awards.
I mean, there's one other person that I have to sort of mention, and he's retired now, but sort of another Shlomo, but Shlomo Lustig. And Shlomo, when I was at Washington University, would come, as a visiting scientist, and really contribute a lot to our work on these arboviruses, these mosquito-borne viruses. And yeah, no, he was an amazing sort of viral pathogenesis person. So I guess he was kind of like the founder. Okay, everybody.
Okay. All right. Great Thank you so much.
Sure, my pleasure.
Bye-bye. Shabbat shalom. Kulam.
Yeah, shalom.
Bye.